Hello and welcome to Nikolai's Genetics Lessons, and today we are going to talk about definitions of different type of mutations of the DNA. And here is a word bank, so we have to choose among these words in order to answer these 4 questions. And the first question is mutation in which purine is substituted with pyrimidine. So what is a purine? What is a pyrimidine? Take a look. You just have to memorize one word, which is cut, and you know that cytosine base pair with guanine and thymine with adenine, and we can find uracil only in messenger RNA, but it's also going to base pair with adenine. So we can circle these 2 bases. They're just going to be different versions of the same base. You also have to know that cytosine has 1 ring structure of the base and uracil and thymine. Hence, we use this word cut in order to memorize that these 3 bases has only 1 ring structure of their bases. But guanine and adenine has 2 ring structure of the bases, and this allows when these bases would align on the different, of course, strands of the DNA, we are going to have the same Ys between bases. This is going to be a bridge of the 3 rings. And we are not going to find, for example, a bridge with only 2 rings structure or with 4 rings. So this rule, which is Charkov's rule, gives us proportion of the cytosine and guanine, which is going to be the same, and thymine and adenine, which is always going to be the same in double-stranded DNA. Next step would be to memorize that this short version of the bases, cytosine, uracil, and thymine, we call with a long word, which is pyrimidine. And those with 2 ring structure, which is bigger, but on the contrary, we call them with word which is shorter, purines. So let me circle. So this is going to be purines, and this is going to be pyrimidines. Now, if you check all the answers, probably you will pay attention to these 2 variants of the mutations, which is transversion mutation and transition mutation. And many students always mess up these 2 variants of the mutations. And here is another mnemonic technique for you. So transversion, first letters, so let me underline, transversion is the same like in transvestite. So when person, for example, male, change its gender to female. So this is something very abnormal, but transition, on the contrary, is something normal. For example, when boy change to man over the time. So transversion mutation is going to be more radical mutation in comparison with transition mutation. For example, if any of these bases would change with itself, we call this transition mutation. Or for example, if one purine would be substituted with another purine, we call this transition mutation. But if one pyrimidine would be substituted with purine, then this is going to be transversion mutation. Or for example, if purine would be substituted with pyrimidine, again, this is going to be example of transversion mutation. In other words, when base with 2 rings would be substituted with base with 1 ring, or a base with 1 ring would be substituted with a base with 2 rings. So our answer to the first question is going to be answer H, or term H. And second question, mutation caused by the addition of a base in a protein coding region. Protein coding region we call gene. So imagine that this is gene. Of course, DNA is going to be double-stranded, but we don't need second strand. We just need 1 strand in order to demonstrate what kind of mutation this is going to be. So imagine now that we have A, T, C, C, ATGA-ATTC sequence. I just took a random sequence from my head. And as you know, 3 nucleotides specify one amino acid. And we call this codons. And let's take a look. Here's going to be our first codon, second, third, and fourth codon. Take a look at this codon table. It is based on the messenger RNA, so on the RNA, and instead of thymine, we see here uracil. But we also can make the same table where we are going to have thymine if it's going to be double-stranded DNA, because double-stranded DNA specify sequence of the messenger RNA. Specifically, coding strand of the DNA is going to be the same as the sequence, with the only difference that in DNA we are going to have thymine, here we are going to have uracil. It would be more appropriate to say that template strand of the DNA specify the sequence of the messenger RNA, but other strand of the DNA is going to be coding strand, and it's going to be complementary to the template strand of the DNA, and the sequence is going to be the same what we see here in this codon table. So take a look. For example, this sequence specify one amino acid, and this sequence specify another amino acid. Now let's take a look what's going to happen if, for example, we are going to have insertion of one nucleotide here in this place. Let's take a look. For example, let's say this is going to be thymine. The first trinucleotide or codon is going to be the same, and it's going to code for the same amino acid. But second is going to be different. If before we had a sequence CAT, now we have sequence TCA. And of course, this is going to be different amino acid. This DNA specify again messenger RNA, and messenger RNA specify sequence of the amino acid or sequence of the protein. And as you see from this place and on, the sequence of the amino acids are going to be different. So we are going to get completely different protein, and we call such mutation Frameshift mutation. So our choice is G, and I also want to emphasize that when we have a Frameshift mutation, it doesn't mean that we are going to have the same length of the protein. As you know, proteins sometimes have hundreds and thousands of the amino acids, but if we will have such Frameshift mutation, what's going to happen? For thousands of the codons, we are not going to see stop codon. We have 3 sequences which specify stop codon. So this is going to be one sequence, this is another, and this is yet another. So out of 64 variants of the trinucleotides, 3 would specify stop codon. That means that 1 out of 20 roughly would specify stop codon. And that also means that if before this sequence coded for protein which had thousands of amino acids, now we estimate that in about 20 amino acids we are going to find a stop codon, and this is going to be also very short version of the protein. So this is also going to be a type of the nonsense mutation, which means that there is no any sense in such a mutation, and this is going to be completely non-functional protein, especially if such insertion would happen early in the sequence of the gene. Now let's check the third question. A base change resulting in a stop codon. As I said earlier, we just can change one base in this codon, and right away this is going to be just by chance stop codon. So we call this nonsense mutation, and also Frameshift also cause nonsense mutation. Maybe not right away as an example with nonsense mutation, but 20-30 codons down the stream. A base change resulting in a stop codon is a nonsense mutation. And the last type of mutation, which we are going to discuss today, type of mutations that cause mutant sectors to appear on the flower petal. This is easy question. You just have to understand that there are only two types of tissues. One is going to be somatic tissue, and another is going to be tissue which produce eggs and sperm, produce gametes. And the second type of tissue we call sex tissue, or also we call germline tissue. So, take a look. Flower is sexual organ, but petals of the flower actually doesn't participate in production of the gametes, which in animals are going to be sperm and egg, and in plants it's going to be egg and pollen. So we are going to choose answer E. Such mutation is going to be a mutation of the somatic tissue. Answer E. One more time, if we compare with animals, think about it. For example, penis doesn't produce sperm, it just conducts sperm, and vagina doesn't produce eggs. So sex organ is not necessarily consist of the sex line cells or germline cells, only small tissue of the sex organ produce gametes. And this is all for today. Subscribe and see you in the next video. Goodbye.